let's go for the third part of the discussions that we have today and talking about post-birth brain and then the extension of the development that we need to discuss about today. In the third part, we will discuss about the next steps after the child being born. In a newborn child from the age of 0 to 3 months old, 6 months old, 15 months old, and an adult, as you have seen, the amount of dendrites that we have inside the brain and for each kind of cell would just significantly increasing over the time, and we have more connection and more synapses, and they contribute to the process of brain development after birth. So the numbers of neurons would remain almost the same around after that period of development, but the number of connections would just go up and up in a daily basis. The fourth process that would contribute to the brain development would be what we call myelination. Okay, so as you can see here, these are just the same results in two different sections. This one is, could you tell me about the cuts that we have here? This one is, what type of cut? Is this a horizontal, coronal, or sagittal? This one? The first one? Just write it in the chat space. Sagittal or axial or... Yeah, this is coronal. And what about this one, the second one? This one is sagittal. As you can see here, in really early childhood, we do not have very much myelination, and myelination would be happening almost after childbirth. So before childbirth, we do not have all these myelin development. And what is this technology that we are using here? Do you know the name of this specific imaging method? Do you remember that? What is the imaging method that we are using here? Yes, we are using DTI. So DTI, as you can see here, we have an evolution in terms of the amount of myelination that we have inside the brain. So that is the process that we have. Okay, this is one of the most beautiful figures that I really love in terms of understanding how brain would work after childbirth. So as you can see here, this is the process that we discussed about neurulation, proliferation, and migration. So we discussed about these processes before, okay? So now we have all neurons in place. In the first few months, the numbers of synapses would just go up and would be skyrocketing. So you can see that they just go up in the first few months of life after birth. And then they would start to just go down. So why they go up and why they go down? The phase of going up would be related to provide potentials for connection. So we just provide all these connections. So we just extend the numbers of connections that we have. And the higher number of connections would provide higher numbers of potentials that a kid would be able to have. And then over the time, based on the environmental exposures and trainings, let's say you develop lots of connections, and over time you realize that it seems that some of these connections are not good connections, and you just keep few of these connections, but you try to just empower those important connections that are more important for you. So we call this process what we call pruning. And this is exactly what we do with plants. So we try to just reduce the number of branches that we have in the trees to be able to have more strong, let's say, stalk or the main body of the tree, to be able to have a stronger kind of branches in the tree that's called the process of pruning. So we start to have an increase in the level of, let's say, cortical thickness, and then we, over the process of maturation, in the process of pruning, we lose some of these connections and we keep the connections that are more important for us based on the experiences that we received during early childhood. So all the experiences in early childhood are important to define, okay, what are these potentials that would remain in place and what are the potentials that we lose forever and we will not be able to bring them back. Whether there are some specific language capacities that we can have at the age of few months old or few years old, or they could be some, let's say, social interactions or potentials that we have to connect to others that are there at the age of 3, 5, 6 years old, but if we do not use them, technically we will lose them. So that is the rule that we have in this pruning system. And the other important part in this process is that this development milestone, whether we go up and down, would be completely different for different parts of the brain. And we have prefrontal cortex being really delayed in this process because of the importance of all these experiences that we are making during, let's say, the first 7, 8, 9 years of our life, and then pruning the system in the next couple of years to be able to reach to a completely advanced, well-established connections that are going to shape our personality and our kind of characteristics over the time. While this process is happening, we can see kind of lots of vulnerabilities in this system and how all these early childhood stressors or early childhood exposures would help kids to develop a totally different brain that they can have. As we discussed before, we still have some probabilistic epigenesis pathways. So we still have specific lines, but based on this process, we can go from line 1 or 2 or 3 to something that is going to shape who we are in adulthood. This process would be happening in terms of folding of the cortex. I told you the process of gyrification. And then, as I told you, the gray matter would be reshaping in terms of the maturation over the time. And as you can see here, white matter would start to develop in terms of the myelination, as we have discussed. As we have discussed before, as you can see here, we had just a little bit of brain being myelinated, but over the time, all the white matter parts of the brain would get myelinated over the time. And then we can get to questions regarding, okay, if we ask about the relationship between things like different cognitive performance measures, like let's say IQ and cortical thickness, how we can measure the relationship between, if we have a thicker cortex or thinner cortex, is there any relationship between having a thicker cortex or thinner cortex related to your IQ or level of, let's say, cognitive performance that you have? People have done that in a sample of subjects. They realized in most of the areas in the brain, the correlation is, I should put 0 here. So 0 to 0.4 to minus 0.4. In most of the areas, in almost all the parts of the brain, the correlation was close to 0. So there was no correlation between the level of cortical thickness and IQ. Okay? But if they just select the group of really young children in their really young, let's say, ages, you can see here in many parts of the brain there is a negative correlation. So something like this. If it is IQ, this is the cortical thickness. If I show it like this, you can see here the higher cortical thickness, the lower IQ. So that is the relationship that they can have. It means that if you have a really thick cortex, it means that your cortex is not still in the process of pruning. And the higher cortical thickness, it means that you have lower IQ because your cortex has not started to go to the pruning phase. So you still have a really thick cortex while you need to start to just reduce the cortical thickness to be able to function better. But if you go to the late childhood, then the correlation would be positive in most of the areas, because when your cortex get developed, in the developed cortex, let's say if it is the IQ, if it is the cortical thickness, the higher cortical thickness, it means that you have better IQ. So you can have totally different relationship between cortical thickness and IQ in different kind of periods of life because of the dynamic process I showed you before in terms of going up and going down. So it depends on where you are located in this pathway and how these items are related to each other. And it is the case that when we go to early adolescence and adulthood, things would just get more complex in terms of how these things are related to each other. And do not forget that even when we are talking about the correlation between cortical thickness and IQ, you need to be careful that we are talking about, in the best case scenario, R of 0.4. It means that the R2 would be 0.16. It means that you are just explaining 16% of the variance in the IQ data by cortical thickness, which is still really good. But most of the studies, they realize that, in reality, there are just very few areas that have that level of correlation. And if we control that correlation, and if we control for multiple comparison errors, at the end of the day, what you get usually, their level of correlation is something like 0.2. And 0.2 means that your R2 would be 0.04, which means that you are just explaining 4% of the variance with the cortical thickness and the structure. And that is what we know, that right now, if you give me the cortical images of a kid and you ask me to estimate the amount of IQ that they have, I can just explain 4% of the variance, which is not really high. So you need to be really careful about that. And there are specific considerations on how to use brain imaging to be able to estimate the cognitive performance of kids. Let me show you one thing that people are trying to do. There is a large study that we called ABCD project. We'll discuss about ABCD project before. This is an adolescent brain cognitive development. This is a large study with, I think, over 35 sites across the US. One of the sites is our center here. We have recruited something like 700 kids so far. And overall, they are recruiting 25,000 kids at the age of 10 years old and follow them up to age of 20 years old to see how their brain would be developing in the process of time. And we already have databases. I think 5,000 is released, 10,000 is released as well. So I think 5 or 10,000 kids, we already have the database publicly available outside in ABCD website. And there was a challenge, something like a competition last year, discussing about, can you predict the IQ of kids based on the baseline data that we had at the age of something like 10 years old, and using the structural brain imaging to explain the level of variance in terms of the fluid intelligence or IQ. This is the page of the challenge. It goes back to November 2019. So the question was how much of the variance they can explain and what would be the best performance of just using the images. And so the process is they give you something like, let's say, 4,000 images, okay? And for these 4,000 images, they also give you the IQ of these 4,000 kids, okay? So you develop an algorithm to be able to predict the cognitive performance or IQ of these kids based on their brain images, okay? And then at the next step, you submit your algorithm. And then there is another database of 1,000 kids that they test your algorithm in that database to show that your algorithm could predict the IQ based on the MR images in an unseen database that the algorithm has not seen before. This is a kind of a classic, let's say, prediction analysis pipeline for those who are doing machine learning. So it's a really classic pipeline. And people try to do that. And probably can't, yeah, I think that people have, yes, this is the amount of, the teams that they have been successful to do that from UCL, Iowa, Purdue, different teams around the world. And the mean square error, the lower mean square error, it means that they have better performance. You can see that the first few groups, they have really close mean square error that they have reported. And then there's a validation, as you can see here. This is the validation phase that they use unseen database. As you can see here, the unseen database was 415, as you can see here. Let me see, I'm not sure how many were in the actual database. Okay, I probably can find that somewhere around here. But yeah, that is and then that was the final program that different teams presented their pipeline. So it's quite amazing in terms of how this process is being organized. Wes is one of the faculty members in our center. He's in in UCSD, but he's also working with us, our institute as well, and Suzanne Taper, and she's one of the leading people in the field, and Ehsan is involved on that project as well. So it's quite an interesting kind of a challenge. So you can see that people are trying to use these databases to be able to predict. And the bottom line message is the success rate is something like 5 to up to 10% in the best case scenario. So it's still really hard to do that. For those who are more interested to know, I should say that when you go to the extremes, let's say, especially kind of lower extremes, those who have IQ of, let's say, under 90 or under 80, that percent of signal that could be explained by brain images would be higher, up to 30–40%. But when you go to the people who are in the normal range of something like 100, 110, 95, that range, the amount of variance that the brain images could explain would be much lower. So that is the way that we know that there are definitely advantages of using these brain imaging methods, but probably not in normal developing kids as much as we can use these measures for those who have lower level of IQ or they have some problems in their development process because they have more extreme, let's say, variances in terms of the both functions and also structure, and that would make the prediction pipeline more successful. Okay. So, and there are some papers, you can find some papers that people have reported on the ABC Neurocognitive Prediction Challenge 2019. They're already in the process of publication. So you can find some of the successful groups. They have published their pipeline, their results. So you can go and find some of those kind of interesting findings there as well. Okay. And then something like the last piece for this talk, for this section of the talk. These are resting state fMRI networks in kids, okay? There are some large-scale networks that you see here. Could you name these networks, the networks that we can see here right now? What is the name of network C? Do you know what is the name of network C here? You can type it. Yep. This is DMA. And what about B? Yes, B is visual. And what about A? Yes, somatomotor. What is the network that is missing here? What is the network that is missed here? Yeah, it seems that ECN is not here. So you can see that if we do large-scale brain network exploration in kids, there are some networks that are not still developed. So even in the large-scale networks, there are some networks that should be developed. And then the process of development, we need to have these networks to be functioning. Okay, so that is the end of this part.